So welcome everyone. Let us look at the cell wall, cytoplasm, and the cytoplasmic organelle. You can also use the word organelle without this one, but this cytoplasmic organelle means that the organelles are actually inside in the cytoplasm. So we are going to see what the cell wall is, what is it made up of, and what are its functions. Then we shall look at the cytoplasm. We will see what makes up the cytoplasm, and we'll see the functions of the cytoplasm. And then we'll look at the different organelles that are found within the cytoplasm. So this is going to be quite lengthy, about 1 hour probably. So please make sure that you are really set to follow this conversation from start to end. Okay? So we start with the cell wall. The cell wall is actually only found in plant cells. You cannot find the cell wall in animal cells. So the plant cells and the bacterial cells, actually, or the bacteria are the ones that have the cell wall. So plants and the bacteria. The animals do not have the cell wall. Okay, so the cell wall is this here, the outermost covering. That is the cell wall. Okay, it's here. That's the cell wall. That's the cell wall. So this cell wall, they are in contact with the cell walls of adjacent cells. So when we say adjacent cells, we are meaning the next cell. They are closely packed, right? So one cell wall, a cell wall of one cell is connected to the cell wall of the next cell or the adjacent cell, and there is usually a communication. That is, there are channels that connect between the cell wall of the adjacent cell wall. So you have those connections being called plasmodemata. Okay, plasmodemata. Those are the connections. I don't know if I've pronounced that actually right, but it is basically just like the way you have the stomata. They are actually called. Okay, they are just actually what's called plasmodesma. That is one hole that is actually connecting, or one pore that is connecting the cell wall of one cell to the other. But when they are actually many, then we call them plasmodemata. I'm pronouncing that correctly. Okay, so the cell wall is basically playing about three functions, major functions. One, it provides protection. Two, it provides structural support. And then three, it provides it basically maintains shape of the cell. Okay, like I said earlier on, the cell wall is found in plant cells, bacterial cells, and also fungal cells. So that means animal cells do not contain the cell wall. That's something that we need to note there. So the cell wall actually has layers, the primary layer and the secondary layer, and these layers are actually having several components in there. We have cellulose, which is very abundant in plants, and then that is one, and then also another substance called lignin is also there, and then the third component called the pectin is also there. Okay, so three major functions: support, maintaining shape, and also issues to do with maintaining the integrity of the whole cell. Okay, so support is what you can term as mechanical strength, maintenance of shape. We have looked at that. The cell wall is actually very tough. This means that when you have a lot of water entering to the cell can swell, yes, but it cannot burst because the cell wall is actually tough because of the components that we have talked about. The cellulose, the pectin, and others. We play an important role in maintaining that integrity so that they don't burst. And then, of course, it being tough, the cell wall being tough, it means that you cannot have pathogens easily entering and causing infection in the cell. Okay, so that is the next role there, protection. And then it also has pores that we have just seen, the plasmodesma here. These pores allow some material to move from one cell to the other. The cytoplasm, the whole this fluid that you see here, the yellowish fluid, that is actually the cytoplasm. It's that space that is filled with the fluid. Okay, it surrounds the nucleus and also contains the organelles, what we earlier on called the cytoplasmic organelles. So in the cytoplasm, you have the organelle. You find their mitochondria. You find their chloroplasts. You find their lysosome, the reticulum, the reticulum system. You will also find there a number of chemicals, carbohydrates, lipids, some proteins, there a number of things. So you can see here all these are the things that you can find within the cytoplasm. It's a fluid, and that fluid contains organic and inorganic chemicals. Okay, what we are referring to as materials here. Okay, and then chemical reactions actually do take place within the cytoplasm. Okay, issues to do with the respiration, some of the reactions actually happen within the cytoplasm before they happen in the mitochondria, for example. Okay, so largely the cytoplasm contains organelles and has that fluid that contains all these chemicals, and you also have chemical reactions happening in there. So the organelles that we have in this image, we have the vacuole, this here, that's the vacuole, and then you have the mitochondrion there, and then you have the chloroplast there. So you have this is the chloroplast, this is the chloroplast, this is the chloroplast, this is also the chloroplast. And then you come to the nucleus with its nuclear envelope here. The nucleus also has pores. We call them nuclear pores. Okay. And then you have the nucleolus, this part here. Okay. And then you have some microtubules. Okay. Here, the ones that actually play an important role in what we call cytoskeleton, okay, in supporting the cell vector, and then the endoplasmic reticulum also there. So those are things that are contained within the cytoplasm. So organelles are those small structures that perform specific functions. They have specific structures that allow them to carry out this function. And in the previous image, this image here is for a plant cell, and then this image here is for an animal cell. So whether it is a plant cell, whether it is an animal cell, all of them have organelles, tiny structures with with the specialized structures that allow them to carry out specific functions. So all the organelles that we are going to mention about, or talk about, actually have specific structure and specific function. So they are quite many. Here we have listed a few of them. So let's just go through these that I've listed. The mitochondria, that is the plural of the mitochondrion. They are very important in generating energy in the form of ATP. And then the chloroplasts, they contain chlorophyll, so they are the ones that pump the light energy and therefore play an important role in the process of photosynthesis. And then the ribosomes, these ones play an important role in protein synthesis. And then the Golgi apparatus, very important in concentration and packing of secretion. And then the smooth endoplasmic reticulum. So these ones are very important in lipid and hormone synthesis. And then the lysosomes, these are very important in degradation of macromolecules. And then the nucleus, which is basically the control center of the cell, controls most activity, but also contains the genetic material. And then we have the centrioles. These are also very important during cell division, okay, especially in the organization of the apparatus. So these are few that we are going to look at each of these organelles in detail and see what other functions do they really carry out and how do they really look like. Okay, so let us start with the nucleus. This here. You can notice that the nucleus in the plant cell is actually towards the peripheral. Okay, it's not located in the center. And then when it comes to the plant cell, the animal cell, if we can go there, animal cell, the nucleus is actually more to the center. Okay, that is something that we can note there. We are saying that the nucleus is actually the center of control. So all activities that take place within the cell, they are controlled by the nucleus. It is a membrane-bound organelle in eukaryotic cells. Okay, there is a membrane around it, and that membrane is what we term as the nuclear envelope. Okay, the nuclear envelope also contains what you can see here, the nuclear envelope here. These are around the nucleus. They have holes in between there, what we call the nuclear pores. They allow movement of the material across. So the nucleus has the nuclear envelope that encloses the nucleus, and the nuclear envelope has some holes which we call the pores that allow exchange of material. That means some material can get out of the nucleus into the cytoplasm, or from the cytoplasm into the nucleus. Okay, the genetic material is actually inside the nucleus. Okay, the genetic material is inside the nucleus. Unlike the genetic material in the prokaryotes, the genetic material in the prokaryotes is actually found in a region that we call the nucleus. Okay, the nucleus where we find the genetic material. Okay, so the nucleus contains the genetic material. It controls all the chemical activities that take place inside the cell, and then also, since it contains the genetic material, it is responsible for carrying and even storing the genetic information. Protein synthesis also has the part where part of the stages of the protein synthesis actually happen in the nucleus. Okay, and then the process of cell division also is controlled by the nucleus. Okay, so this is basically, in summary, to say that all chemical activities that happen within the cell are controlled by the nucleus. Okay, containing the genetic information of the genetic material, controlling the process of cell division, playing an important role also in the process of protein synthesis. Actually, protein synthesis is initiated within the nucleus. Okay? So the nuclear envelope is what I've just explained, what encloses the nucleus. Therefore, it controls what enters the nucleus and what actually goes out of the nucleus. Like the plasma membrane, it is actually selectively permeable. Okay, that means that doesn't allow anything to just enter. Okay, it is very selective. Okay, contains the nuclear powers, which are the holes through which this material moves. Okay, and basically the membrane that separates what is inside the nucleus and what is outside, that is the cytoplasm. The chromatin and the chromosome, these are actually contained within the nucleus. Okay, these are actually contained within the nucleus. When we talk about the chromatin, these are coiled inside the nucleus. They are, if they are tightly coiled, then we actually call them the heterochromatin. So if you apply a dye that we call a stain, you apply a stain, it stains intensely, okay, and it is located closer to the nuclear envelope. Okay, when it is not tightly coiled, then we actually call it the ochromatin. Okay, that's EU here, appearing there. Okay, and this does not mean that it is actually genetically inactive. Okay, this ochromatin is actually genetically active during the interphase, what we usually come as the resting stage of the cell cycle, okay, or of cell division. Okay, so the chromatin are actually very active during the interphase. And then we have the nucleolus. Okay, it's a prominent structure in the nucleus. So within the nucleus, let's go back here. So you see this part here. That is the nucleolus. It is inside the nucleus. It contains the chromatin. These are basically coiled chromosomes. Okay, just like what I've just described. Okay, so it appears round and it stains darker. Okay, because of those genetic material that we actually find there. So they play an important role in assembly of the ribosomal RNA. So the nucleolus is very important because it plays an important role in the assembly of the ribosomal RNA. You look at the transfer RNA, the ribosomal transfer RNA, and then also the N or the messenger RNA. You look at all those things during protein synthesis, you see their role there. Okay, so the nucleolus is responsible for ribosomal synthesis. Okay, so let's look at the endoplasmic reticulum. When we talk about the endoplasmic reticulum, we are meaning a network of membranes. These membranes here, you can see them in your images. So these membranes are very important in two things, two major things. protein synthesis protein synthesis or protein metabolism and lipid metabolism. So what differentiates the other is that what differentiates between them are these dots that you see here. These dots are the ones we call the ribosomes. Okay, they are the ones that are responsible for protein synthesis. And because of these ribosomes, we call that the RAF endoplasmic reticulum. And then the other endoplasmic reticulum does not contain the ribosome, and therefore it is small endoplasmic reticulum. So this one is for protein synthesis, and then this one is for lipid synthesis. Okay, so the RAF is RAF because of the ribosomes, and the ribosomes are the ones that are very important in protein synthesis. Okay, but also they can act as the pathway for material transportation, and the fact that you have these ribosomes there it increases the surface area of chemical reaction. And then we come to the smooth, no ribosomes, and then also responsible for synthesis of lipids and also hormones. They can also be used for storage of glycogen and lipids, and also breakdown of chemical substances in the liver, which is basically detoxification. So the smooth endoplasmic reticulum is responsible for three major functions: lipid synthesis, detoxification, and then storage of glycogen. And then we move to the mitochondrion. The plural is mitochondria. Okay, they are the ones we term as the powerhouse because they actually generate the power or the energy that the cells will need to carry out different metabolic processes. Okay, so energy generation in the form of ATP happens in the mitochondria. Okay, so this is the structure of the mitochondria. So let's look at the structure in detail. It has two membranes. We call them double membrane. So the outer membrane and the inner membrane. The outer membrane is what is in contact with the cytoplasm. The inner membrane is what is in contact with what we call the matrix. And then we have some folding that we call the cristae. Here, these are the cristae. We will see that they actually increase the surface area for chemical reactions. It's basically the structural reactions that happen inside that. So inside we have this fluid-like substance, like for example what we can term cytoplasm. That's basically called the matrix. It contains the number of things, the ribosomes. Okay, contains the ribosomes, contains the phosphate granules, the respiratory enzyme, for example, ATP, and ATP synthase enzyme, and then also mitochondrial DNA. Okay, so the mitochondria has its own DNA. That means that it has it is semi-autonomous. It means that it can really pass on the information, genetic information that it has. Okay, so the fact that the mitochondrion have the mitochondrial DNA, it makes them semi-autonomous. And then we have the intermembrane space. So between the outer and the inner membrane, we have a space there, and that space is what we call the intermembrane space. Mainly the function of the mitochondria is to make energy in the form of ATP. Okay, but also it is involved in the synthesis of other molecules. For example, chlorophyll, cytochromes, steroids, and fatty acids. Okay, and then it's very important in storing and releasing calcium ions. Therefore, it plays an important role in regulating the amount of calcium in the cell. So what makes the mitochondria very good at producing energy? Okay, it has the outer membrane porins. These allow passage of molecules that are very important for the process of respiration. Okay, and then those foldings of the inner membrane that we call the cristae, they increase the surface area for more reactions to happen, for more ATP to be produced. Okay, and the mitochondria communicates with what is in the cytoplasm. That means that it can allow some things to enter and other things to go out. And that is very important for it to get the material that it needs for the process of respiration. But also within the matrix, okay, we have enzymes that are very important for energy production. Now we look at what we call the plastids. These are membrane-bound organelles that are found in plants and the algae. Okay, and they are responsible for food production and storage. Okay, so these are very important actually in photosynthesis. So we have three categories. We have the chloroplasts, 1, and then the chromoplasts, 2, and then the leucoplasts, that's 3 there. Okay, so the chloroplasts are the ones that contain chlorophyll, very important in light absorption. And then we have the chromoplasts, they also contain pigments for pollination and cytoplasm, and then the leucoplasts are very important for storage of fat, proteins, and lipids. Our focus is going to be on the chloroplast. Okay, perhaps you look at the structure of the chloroplast again in what we call the process of photosynthesis. But let's go through the structure in here. So we are saying that the chloroplasts are found in plant cells and some green algae that actually carry out the process of photosynthesis. So the plants are not the only organisms that actually carry out photosynthesis. We can also have what we call the green algae carrying out photosynthesis. So the light is sparked by the chlorophyll that is contained in the chloroplast. So without the chloroplast, you cannot have the process of photosynthesis actually happening. That means the plants cannot make their own food using light energy to form chemical energy. So it's also like the mitochondrion. So the chloroplast have 2 membranes, the inner membrane and the outer membrane. Okay, the outer membrane is permeable and actually smooth. That means that being permeable allows certain molecules and ions to go into the chloroplast. Okay, so you can see these are the 2 membranes that we are talking about, the inner and the outer membrane. The inner membrane is actually, the outer membrane is smooth and also is permeable. That means it allows some ions to go through. And then the inner membrane is less permeable and contains what we call transport proteins. The transport proteins are the ones that allow some protein to move across this membrane. Okay, so that means that because it has control, transport protein, it can control what comes in and out of the chloroplast. Okay, and then of course there's that space between the outer membrane and the inner membrane. So we call that the inter-membrane space. It's very narrow and it acts as a barrier between the cytoplasm and the chloroplast interior. The fluid in there, remember the fluid, where you have the organelles floating, where we have the organelles in the cell, what we call the cytoplasm. We call it the cytoplasm, right? And then we move into the mitochondria, where we also saw the matrix. And now we are coming to the chloroplast, and now we are seeing something different, called the stroma. Yes, you see the difference there. Okay, so it is a dense fluid within the chloroplast, surrounded by the inner membrane, and you have all the enzymes that you need for the process of photosynthesis. Okay, so it contains all the enzymes that you need there. And of course, with the time when you look at the process of photosynthesis, you know that you have what we call the light independent phase and then the light dependent phase. And that's where you see the role of the different enzymes and the role and what reactions actually happen within the stroma. Okay, and then we have the alkaloid membrane system. So within the stroma, you have a network of membrane-bound compartments. Okay, they're the ones that we call the alkaloid system. So basically, let's go back to that image again. You see this connection here. This one is connected to this one is connected to that. This all these complicated things here. That is the alkaloid system that we are talking about. So what does it contain? We have the alkaloid lumen, basically the inferior space. When we talk about lumen, that space inside the tube-like structure, that's the lumen. Okay, so we have the alkaloid lumen. That means if you pass across, you see that space there. That space is very important because it contains what we call the protons. And these protons are very important during the life stage of photosynthesis. And then we have the grana. You have a stack of the alkaloids. When you have them being stacked on one another, this one here, you see this part there, that's the grana. When you have this here, it's one grana. When you have several of them, this, that, and that, that's when you have the grana. Okay, so we have the lumen, we have explained that, we have the grana, and then the alkaloid membrane, basically the membrane that's covering all these alkaloid systems. They are the ones that contain chlorophyll. Okay, this is very important in fact in life. Okay, in plants we don't only have chlorophyll, we can also have other pigments. Okay, other pigments are also can be embedded within the alkaloid membrane. Okay, though of course chlorophyll is the primary pigment that plays an important role in photosynthesis, but there are also other pigments in there. Those other pigments, for example, the carotenoids, and then xanthophyll, and also some of the pigments found in green plants. So if we ask you about examples of plant pigments, then you should be able to mention chlorophyll, you should be able to mention about the carotenoids, and then the basophil, you should be able to mention them. Like the mitochondria, the chloroplasts are also semi-autonomous. That means that they have their own mitochondrial, and I mean they have their own DNA. So they have their own DNA. That means that they can actually produce their own protein independently of the whole cell. When we say an organelle is semi-autonomous, that means that it has some power about doing its own thing, regardless of what happens in the entire cell. So when we say that the chloroplasts are semi-autonomous organelle, it's because they contain this DNA. That means that they can carry out some function in their own. And then the plastoglobulins, or plastoglobule structure, plastoglobule. These ones, they contain, these are lipid-containing structures found within the stomach. So that means that they're very important for lipid storage. So photosynthesis primarily happens in the chloroplast, but also since you have some ribosomes within the chloroplast, that means that the chloroplasts are also involved in protein synthesis, okay, and also some starch and lipids and liquid in the chloroplast. The chloroplasts can do their function because they have all these pigments, the chlorophyll and the other pigments that I've just mentioned about. Two, that they have these enzymes that are very important for photosynthesis. And then three, you have it being permeable to some ions and material to enter into the cell, to enter into the chloroplast for use during photosynthesis. And then the grana, they increase the surface area for which photosynthetic reactions can actually happen. The pigments I've talked about them, and then we also have some proteins embedded within the grana that also are very important in what we call photosynthesis. And their chlorophyll molecules are very important for harvesting more light or trapping more light. Presence of the enzyme is something that I've explained. Being semi-autonomous can be called self-replicative because they have their own DNA. They can control some of the processes that happen. For example, the process of making certain proteins. Okay, so they contribute to genetic inheritance and also enable the new cells to have the necessary machinery for photosynthesis. Having ribosomes means that they can actually play an important role in photosynthesis. So we go to the vacuole. We are saying that the vacuole is located in the center for plant cells, but for animal cells, they are actually usually very small and temporary. They are used for storage and transport. But for plants, they play an important role in maintaining structure, maintaining cell structure of basically plant support and storage of nutrients and even minerals. The vacuole is surrounded by a membrane that we call the tonoplasm. Okay, surrounded by a membrane that we call the tonoplasm. So the functions of the vacuole: support, okay, contain food material and minerals, and then also very important in what we call photolysis of dead cells. Okay, the cells, if cells actually die, they have, if the cells get old, they have to be killed. Okay, that is what we call photolysis, and maybe we can look at something about it shortly. And then osmotic regulation, what goes in and out of the cell is controlled by the vacuum. Okay, and those are issues to do with osmotic regulation. You cover osmotic regulation under transport in plants. Okay, so we are saying that when cells get damaged or when they get old, they are destroyed. Okay, so autolysis is self-destruction of the cell through action of its own enzymes, just like suicide. The cell kills itself. Okay, that is autolysis. Auto, self. Lysis means to break. Okay, so how does it happen? The membrane that is around the vacuole ruptures, and when it ruptures, it releases this enzyme that now can digest the entire cell, leading it to die. Okay, that is autolysis. The other function that we can also mention, especially for vacuoles in plants, one is that they provide that support, and that support basically comes when you have fluid entering into the cell, and then that cell expanding and exerting pressure on the cell wall, and that is how the plants, actually habitat plants, are able to stand upright. Okay, because of the cell vacuole. Okay, but they also, that expansion is also actually contributes to growth. Okay, that expansion is also contributes to growth. And then the vacuole also contains some pigments that are very important in flower color, in fruit color, and also in the color of the buds. Okay, those are the anthocyanins. Anthocyanins are the pigments that are very important. Okay, if we have the flowers colored, then we have increased chances of pollination and therefore seed development and later on the flower. So we move to other organelles that we call the ribosomes. Yeah, so ribosomes are very important in protein synthesis. They have the larger subunit and the smaller subunit, or the small subunit. The ribosomes are found in both prokaryotes and eukaryotic cells, and also in the cytoplasm of these cells, but also in the mitochondria and also in the chloroplast. We also find ribosomes there. We have ribosomes that are larger, and we have those that are smaller. The ones that are small are the ones that we call the 7th S ribosome, and then the 8th subunit ribosome there. Okay, both in prokaryotic cells you find the smaller ribosome. In eukaryotic cells you find the larger ribosome there. Ribosomes are very important for synthesis of protein. So we have the construction of polypeptide chain happening on the ribosome, and also in the translation of mRNA into protein, and then formation of those bonds between the peptides also happen on the ribosome. Lysosomes are organelles that are enclosed by a membrane, and they contain digestive enzymes. That means these enzymes can digest or degrade foreign material, or even degrade the cell itself. Okay, so the lysosomes can kill the cell, what we call self-digestion of the cell, the process that we call autolysis earlier on. And then these enzymes can also degrade some internal structures within the cell, and that is what we call autophagy. And then heterophagy is when these enzymes basically digest external material. Okay, so the lysozymes are very rich in these digestive enzymes. We can also call them hydrolytic enzymes. Okay, they have to be in position not to digest any cell anyhow. Okay, so that means that what we talked about, cell-to-cell recognition, is very important, that you don't have unnecessary autolysis happening. And then we move to what we call the microbodies. These are small membrane-bound organelles found in eukaryotic cells. Okay, they're involved in several processes, and we can have one example: the peroxisomes. These ones break down hydrogen peroxide in the liver and the kidney cells. And then we have the glycosomes here, and then we have the glycosomes. The glycosomes containing the glycolytic enzyme found in some protozoa. Okay, so you can notice from here that these microbodies are important in metabolism. Okay, metabolism or breakdown of hydrogen peroxide, break or conversion of fats to carbohydrates in the making seed, and then also the glycolytic enzyme found in some protozoa. The Golgi apparatus, these are disc-shaped structures that consist of flattened plate-like compartments we call the cristae. So the cristae are those flat-like compartments. Okay, the Golgi apparatus is very important in assembly of molecules. It's basically like packing and then transporting. They pack and transport, what we explaining there. Okay, so they are very important information of liposomes, because when they break off, they contain these digestive enzymes that we have just talked about. So Golgi apparatus are associated with secretion. Okay, secretion such as hormone enzymes, and also in cell wall formation, especially with plants. Okay, so that basically marks the end of this presentation. It has been a lengthy one. I hope you have picked one or two things.